All right, so welcome to the last week. Although it feels like the last week, it feels like it's been the last week like forever in some sense, seeing as how we've not been at school. But anyway, so we're at nucleic acids. And we pointed out that subunits were nucleotides, and then the subunit had sub-subunits, if you will. Nucleotides are made of three different things: a phosphate group, a pentose, meaning 5-carbon sugar, either ribose or deoxyribose, and nitrogenous bases. Adenine and thymine always have to go with each other. When they're together, they have to go with each other, except in RNA, then adenine would go with uracil. I'm not. Cytosine always has to go with guanine when they're together. Cytosine can't go with thymine, adenine, or uracil, and so on. Pointed out that there were two subgroups: deoxyribonucleic acid, or DNA, which you've heard all about. Two anti-parallel strands, and you remember that anti-parallel means parallel but running in the opposite direction. And it tends to arrange itself into a double helix, or twisted and twisted again, wrapped around in a coil and wrapped around in a coil again. You hear about double helix quite frequently when people, frequently people will use double helix as like a euphemism for DNA. Remember that DNA carries the instructions for every protein that the cell will ever be able to produce. By the way, it tells two things, these instructions. The instructions are either how to make the protein, or when. So two kinds of instructions: how to make the protein, when to make the protein. My favorite example, because it's so short and has such a big impact, is the gene that's called SRY, which produces maleness. It's found on the Y chromosome, and that's why only people with a Y chromosome can be male, because that's where the male gene is. Without the SRY, if SRY gene never expresses, a person would be female. It takes no special gene to make female. Female is the default configuration. So in order to change the development, you need the SRY gene to express. It has to turn on at exactly the right point in development, very early development. It's only on for several hours, like I can't remember exactly, a day, give or take. And if it turns on and everything works right, it develops into male. And if it doesn't turn on and everything still works right, but it didn't turn on, it develops into female. That gene has to turn on at exactly the right time. And it's only on for a few hours, several hours, but not even close to a lifetime. All right, now that brings us to where we left off, which is RNA. RNA, it's complicated. RNA is single-stranded, although how should I say this? Can pair, by pair we mean double strand, can pair / DBL, double strand, temporarily. And part of how it works is by pairing at the right time, but not all the time. Now, there are, forever, we said there were three kinds of RNA. But as we're getting more knowledge, we're discovering more kinds of RNA that are much less common. So these three kinds of RNA are the main types. And you could put the bracket down to all the subpoints, but they're not on the screen right now. So these three are the three main types of RNA. And these together are crucial for gene expression. And gene expression means making the gene's protein. So these ABC, messenger RNA, ribosomal RNA, and transfer RNA together are involved in making proteins. And, you know, remember, proteins do virtually everything that's done actively in your body. And so they're critical, absolutely critical. So this is a very, very important process. So the messenger RNA, or M, notice RNA is caps, messenger is not. If you were to write capital M, capital R, capital N, capital A, Myrna, that would mean, I don't know what it would mean, but it wouldn't be read by anyone as messenger RNA. So this carries the instructions for synthesizing a single protein from the nucleus to the ribosomes. Ribosomes, you remember ribosomes from 10th grade. Ribosomes are where proteins are made. So the messenger RNA carries the instructions from the nucleus where the gene is to the cytoplasm where the ribosome is. And well, in the old days when everything wasn't online and you went to a library, and maybe you still do this, well, at least to some degree, even if you're online, when you want to copy information down, you might use note cards for little pieces of information. Or, and in the old days when you go to the library, especially if you were using the reference section that they would not let you check out, you would write your information on note cards and you could take the note card with you. Well, or another example might be, now different people are different. Some of you might, or your moms or your dads or whoever, might like to keep the cookbooks nice and clean and pretty. So they might copy a recipe onto a recipe card before they go into the kitchen to cook, so that if they spill anything, it messes up the cookbook. I mean, the recipe card and not the cookbook. Well, that's kind of what messenger RNA is like. It's just one specific recipe or set of instructions on like a note card, except it's not on a note card. We're talking figuratively here. That can be taken from the nucleus, that would be like the reference library or the cookbook, to the kitchen, let's say, where you're making, or to the computer where you're writing your paper. And messenger RNA, its structure, they are longer and they are more or less straight strands. They don't have any special folding or anything. Okay, they carry instructions from the nucleus to the ribosomes. Ribosomal RNA, ribosome R, and again, little r, big N, big A, little r. So by the way, this is the pirate RNA, RNA. So this makes up part of the active structure of the ribosomes. The ribosomal RNA is actually in the ribosomes as part of the structure, and these are twisted strands. So the messenger RNA is carrying information from the nucleus. The ribosomal RNA is making up the ribosomes where the proteins will be made. transfer RNA, T, transfer, little t, big R, big N, big A, transfers specific amino acids to the ribosomes from the cytoplasm. So there's these amino acids floating around in the cytoplasm. And the transfer RNA goes out and it finds each transfer RNA has one specific amino acid that it can carry. So the transfer RNA float around through the cytoplasm looking for these amino acids, their own amino acid. When they find their own amino acid, they attach to it and they carry that to the ribosome. They carry that specific amino acid to the ribosome where they're going to be needed to make whatever protein the messenger RNA at the ribosome is making. So these are shorter and they are shaped like a clover leaf, as in they have a loop and a loop and a loop, except they're probably much prettier than that. At least the pictures you can find online are much prettier than that. Now, this is kind of important in the whole structure. They have a three nucleotides which code for the specific amino acid that they hold. So on one end they hold the amino acid, and the other end they've got three nucleotides like maybe cytosine, guanine, uracil, M0, and then that would attach to the complementary base is actually B. Yeah, it's adenine. On the messenger RNA. I guess you don't need to get the complementary business, but they do have this specific three-letter code that instructs for their specific amino acid. Okay, that's the short version. If you take AP Biology next year, or if you go into any biology class in college, they will spend a whole bunch of time on the translation process where genes are made into proteins. Messenger RNA gene is changed into protein. And they'll talk a lot, or I'll talk if it's AP Biology, about messenger, transfer, and ribosomal RNA. Now, there are a couple other lesser known RNAs more recently discovered. And by the way, this is not in the copy of the notes you have. Small nucleotide, or snRNA, is used in RNA processing in prokaryotes. Remember, prokaryotes are bacteria, cyanobacteria, and the archaebacteria. And then there are some even rarer types. Regulatory RNA, and there are several kinds of regulatory RNAs, 3 kinds. Transfer RNAs, not transfer. Transfer messenger RNA, and there are there's an example. This is really fascinating to me. Ribozymes are RNA enzymes. So this is the only real example we have of something other than proteins doing something actively in some cell someplace. And we said that RNA is single-stranded, but there's an exception to just about everything in biology. There is some double-stranded RNA. Now, these, by the way, back up to here, these, you don't have to know. I'm not telling you not to know them. I'm telling you, you don't have to. They will not be on any worksheet or anything this year. They may show up there next year, but you don't care about that. All right. There is a third category of nucleic acids. We had the DNA. That was A. B was RNA. C is independent nucleic acids. Now, for whatever reason, most books don't make, they talk all about these independent nucleic acids, but most books and notes don't talk about them as being a part of the nucleic acid family. So they'll talk all about them and just expect you know what's going on without having said anything. And we've heard of these. We in biology class have already talked about these last year. Single nucleotides with one or two extra. Now you got to think about this. One or two extra means 2 or 3 total, because all nucleic acids have, all nucleotides have a phosphate group. Remember, a phosphate, a pentose sugar, and a nitrogenous base. So if we already have a phosphate and we add 1, that brings us to 2 total. If we add 2, that brings us to 3 total. And you've heard about this one particularly because we talked about this in chemistry. ATP, remember where Indians used to live in the old days. Other ones we didn't talk about in 10th grade, but we will mention in AP Biology, should any of you take that, would be GTP. Sounds like it should be some kind of a car model. It was a GT. I don't know if there was ever a GTP. But we'll talk about GTP, guanosine triphosphate, and GDP, guanosine diphosphate, in AP Biology if you take it. So these independent nucleic acids are used to transfer energy within cellular processes like photosynthesis and respiration and muscle contractions. Remember, I told you whenever you see ATP being changed to ADP, that meant that the cell was spending energy. So that section is in the notes I gave you, except it's handwritten at the bottom very sloppily because that was mostly supposed to be a note to myself to type it in. And I did type it in, but not until after I printed out the notes for you. All right, so I have good news, I guess. On the email I sent you yesterday, I said that we would be doing a Zoom meeting today and tomorrow, Monday and Tuesday. But that's the end of the notes. We will not need a Zoom meeting tomorrow. So this ends the time that you have to spend staring at the screen, listening to me drone on. You will still have a quiz tomorrow. But there will be no Zoom meeting. And other than that, the week will go, like I said, in the email. So we'll see you next year, probably, I hope, maybe sooner. Have a good afternoon.